Welcome to Forethought India's podcast. Today we have with us Julia Schaletzky. She's graduated from Harvard Medical School in 2006 and is currently lecturer at Haas School of Business, UC Berkeley, and also executive director for Henry Wheeler Center for Emerging and Neglected Diseases at Drug Discovery Center, UC Berkeley. Her fields of expertise and research interests are pharmaceutical development, bioentrepreneurship, biotechnology, health policy, public health, and innovation. Today, she will be speaking on drug discovery. The time is yours.
I'd love to talk today about antibacterial and antiviral drug discovery. And this is an area I've been involved in for quite some time. I worked in the biotech industry for more than 10 years, developing medicines against cardiovascular and neurodegenerative diseases, and also did some discovery now in an academic setting with public-private partnership for infectious diseases, such as antibacterial and antiviral medications. I'm particularly interested in antibacterial because it's really one of the big issues of our time, and I would like to walk you through today what needs to be done to develop novel antibiotics. So the discovery process, as you can see, can you see my cursor?
Yeah.
Is a long and quite arduous process. You begin in the pre-discovery phase, which is basic biology research. This is where the academic research is, trying to figure out which new targets could be looked at for therapeutic use, and then you do a big drug discovery campaign, which means testing many different compounds. This can be very large scale, can also be supported by computational methods. And then you enter preclinical phase testing, which means all biochemical cell-based assays, early mouse work, toxicology studies, but also animal models to see if the drug works. So this is a really long, highly iterative process, just super condensed for this presentation, but this can take years to get somewhere, sometimes even longer than that. And once you have a good compound from this discovery campaign that really interrogates all kinds of new compounds, and you have a candidate that behaves well in the animal model, does what you want it to do, is non-toxic, then you get approval by the FDA to do an initial new drug application, and you submit this, and then you can start clinical trials. Then there's in the US, there's 3 phases mostly used. There's also phase 4, but phase 1 is like an initial dose escalation, the first time you give that molecule to humans, and then phase 2 is already can be in sick people. It's a larger trial, but also still establishing toxicity, safety, trying to find limiting a good dose that would allow you to observe the effect that you're after. And then phase 3 is really the largest pivotal trial, looking at efficacy for the first time, truly, at the right dose with the right endpoint, and so on. So this is a really long process, starting from basic biology in the academic setting, and then big discovery, a lot of experiments, animal work, and then studies in humans. And then finally, NDA will be submitted, new drug application, and FDA will decide if the efficacy that is seen together with the tox data that has emerged from the trials, and potential other risks, is arguing in favor of an approval or not. And then there's also post-marketing surveillance that can be added. So the cost of this, if you look at this graph, this is why drug discovery is so difficult and so long, because you need to integrate all kinds of areas of different expertise, different groups, PhDs, MDs, biologists, chemists, regulatory professionals, everybody, and also a lot of lawyers and people who manage the site. Everybody has to work together. And the idea in the beginning has to be good, of course, in order for this to work. So clinical trials can be very expensive, particularly if they're large and lengthy. this can cost. If you would plot this in terms of money needed, the beginning would actually be really small, and the big ticket item would be around the phase 3 trial, and generally in the clinical phase, but phase 3 is the most expensive one. This is also why it's so difficult to really see compounds through, because you need to raise that money. Very few companies can pay out of pocket for a phase 3 trial. So, what kind of therapeutics can we discover in this process? So, the classical pharmaceuticals are all small molecule. Aspirin is shown here in the upper right corner. This means they have a molecular weight of less than 500 grams per mole. There's organic synthesis taking place, medicinal chemistry. This is very well established. We know methods how to make these things in large amounts. India is a leader in this also. They are really cheap to make. We have methods to test purity and make sure we have what we want to have. And it's not contaminated. All this is very well established. So with small molecules, the advantage is that they tend to be orally bioavailable. Not all of them, but you can design them to be. Large scale synthesis possible. They generally cheap and have a good shelf life. You can have pills in your cabinet for a long time and they don't really go bad. So that also means you can stockpile quite well for emergencies or pandemics. And they can traverse cell membranes and the blood-brain barrier if designed that way, and tend to be in the body for a short time. So if you take a pill, generally it's designed that after 1, 2, or 3 days, if you stop taking it, it's completely washed out of your body. Biologics are a new class, relatively new class of therapeutics that contain humanized antibodies. For example, some of you might have heard of Avastin, or other injectable ligands, also like insulin. They're generally more expensive to make, and the QC methods, all this is pretty new relative to organic chemistry. They need to be injected. They don't transverse the cell membrane, and they're highly specific. They can have super high binding affinities. So getting a strong binder is less of a problem with the biologics. But the problem is they normally can't get across the membrane. So your targets have to be targets like VEGFR2 here that protrude out of the cell, and then you can have an antibody bind. They last in the body for several weeks. Often if you take those, you need to do an injection, and self-injection is now available, but you have to inject once a month, and then this is how you dose them. And then in between are large molecules, peptides, really complex molecules, and many antibiotics actually fall in that category. Here is Taxol, a cancer drug, which is from a plant from the Pacific yew tree, and this you can see is very complicated to make relative to aspirin, which is one problem in this area, that a lot of bioactive natural products are very, very difficult to synthesize. So turning this general introduction towards antibiotics, I don't know, maybe you have family members who still remember the time before antibiotics. I certainly do, or when antibiotics were in really short supply after World War II. Back then, any infection you got, and we get infections all the time throughout our lifetimes, could mean significant problems, could mean amputation of limbs, and definitely a lot of pain and suffering, and definitely also death in some severe cases. So, for example, if you have a Staphylococcus aureus infection, 70% of the cases were fatal, which is a significant, really significant mortality. And Streptococcus as well, a third. A lot of children passed away based on bacterial infection, and it's just like more people died than we would think, right? And then what happened is antibiotics were discovered, and a lot of this went away pretty fast until it just came back more recently as we have resistance. But we developed antibiotics really, really successfully in the second half of the last century, as evidenced by the fact that amoxicillin, a standard antibiotic which just is in shortage right now in the US, unbelievably, is actually cheaper than a gallon of milk. So we had so much development and production of really cheap generics. It's the mainstay now of therapeutics. And in a way, maybe you can say philosophically, antibiotics really laid the groundwork for even having a public belief in therapeutic development, because they work so well. I mean, you don't really need for some of these. I mean, you can immediately see a benefit when you take these drugs, and it's very well established. It's kind of it was almost magical to people who have had experience with just bathing, fingers and infected fingers in salt water and putting packs around them and just hoping for the best. So this is a very it was really a new era in medicine when we had antibiotics, starting in the maybe 1950s broadly. But what happened in the meantime? Many of you will know that antibacterial drug discovery has stalled. But we need more of it because of antibiotic resistance. So resistance emerged. Why? It's because we have seen an overprescribing of antibiotics. Also, we use a huge amount of antibiotics in livestock farming. This dwarfs completely what human use is. And also, patients sometimes don't finish their treatments, and we don't develop new antibiotics. So bacteria are extremely flexible, and they can develop a resistance quite easily, much more easily than multicellular organisms can, for example. So the way they do this is they have a couple of mechanisms to get rid of antibiotics very well. First of all, they use a lot of pumps to just pump antibiotics out of their system, but they also have a system to change genetic elements and transfer genetic elements from one bacterium to the other. And that means if one has a resistance gene, it tends to spread really, really quickly, because then if you have the antibiotic present, only the resistant ones will survive. They have an advantage, and like this, they can spread and also spread into the environment. So, for example, if you have a pig farm and you use large-scale antibiotics, and the sewage is then just entering the water stream raw without being treated, you basically taking tons of bacteria into the environment that are now resistant against antibiotics, and those can find their ways into the human environment, and then you have a problem. So this is what happens. We have a lot of really serious threats of antibiotic resistance that I just can touch on very lightly, is because of time, but it's a big problem. So in the past, discovery of penicillin is known to most people based on this fungus. And that was isolated, was really, really a breakthrough by Alexander Fleming. And here's a timeline of the discovery of antibiotics. And this was really, bio mimicry kind of process, because Alexander Fleming found from a fungus, the fungus in its natural environment needs to fight bacteria, and that's why probably it evolved that way. But so more and more antibiotics were discovered over the years, but you can see after the 60s, basically it more or less stalled, and we haven't really had that much discovery in antibiotics at all. A part of this was because we believed the problem is solved, because we had already very good antibiotics, but then resistance emerged, and there's a new focus now on making this work. If you look at the structures of most successful antibiotics, one thing that's interesting is that a majority of them work on several targets. So in a way, it's like a combination therapy in one drug. And this is important to remember, because antibacterial development is inherently challenging. Maybe that challenging that it's actually difficult to find a molecule that only hits one target and will do what it's asked to do. So what are the challenges of antibacterial drug development? The first one is that bacteria have a unique permeability barriers. So the high throughput screening of drug-like compounds is largely ineffective, because the drugs either don't get in, or if they get in, they get immediately kicked out by these pumps, efflux pumps, and you can see that there's often several membranes separating the bacterium from the outside, and that just makes it very, very difficult. You need a molecule that gets through water and gets through oil, so to speak. So it's very hard. And that's one problem. It's not unsurmountable problem, but it's a challenge of bacterial antibacterial drug discovery. The second challenge is that because of this permeability problem, often you have really low cell-based potency of antibacterials. So that means the drugs, you put some in, but you need to use a lot in order to get the effect. So, for example, if you look at a 20 mg daily dose of Lipitor, this slide was made by Ryan Sertz, who has worked in this field for a long time, and I think it's a genius slide because you can immediately see what the problem is. This is your Lipitor dose. It's highly effective and efficacious, and these are antibiotic doses. You know what you need. So you literally have to take like a tablespoon. This is pure compound. There is no excipient. There's no lactose or anything that you typically find in pills. You would just have to take a scoop of this daily. So the problem with this is not just that you have to swallow a tablespoon of powder. The problem is that if you have really high doses like this, your potential for off-target toxicity is pretty high in the body, so you run into all kinds of problems you don't want to have with side effects. And indeed, many of the last-line antibiotics have really severe side effects, and you can lose a kidney or something when you take them. But at that point, you almost dying, so people do it. But this is really one problem. It's because the bacterium is so effective in getting rid of this stuff that you put in the environment that you need to literally drench it in order to get an effect. The third challenge is that manufacturing generally is complex. And why is that? It's because these molecules, if you think about the first slide I showed, are generally large and kind of unwieldy. So if you think about aspirin, that was a tiny molecule, tends to be easy to make, one cyclization. But then if you look at, for example, some common antibiotics like dalbavancin, look at this thing. I mean, this is huge, very, very difficult to synthesize from scratch. Plasmamycin as well, sugar rings there, all sterics, like synthesis, it's difficult. There's different chiral centers. Lipitor, however, is much smaller. This is kind of the standard what a drug would look like. It's relatively small. It has only a couple of rings. So these kind of natural product-based things tend to be much more difficult to make, and that is the problem. You have low yields. Manufacturing is hard, and also there's a lot of rules around sterility testing because you want to make sure you don't have any bacteria hanging around that are already resistant to this, and then you basically bring it into the patient population. So the antibiotics must be segregated away from other drugs, and some antibiotics also from each other. All this increases the cost of development. So this is just an interesting sideline I want to leave you with, that of course every time, even if some well-meaning administrator goes, maybe for safety's sake, we want to do like 3 different facilities and everything is completely separate. This may be a good decision in one way, but in the other way, you also have to consider how much of an obstacle this puts in place for the development of potentially life-saving medicines. And you can make the argument that maybe we should restrict ourselves to only the regulation that is truly needed, and a lot of the gray zone things maybe should take a step back here, because what we need is life-saving new medicines. So that brings me to the fourth challenge. Antibiotic development is really a case of market failure in the United States and also globally. The government should remedy that, but largely fails to do so. So we're slowly getting into this, but this is really a problem I'm going to highlight in the next couple of slides, how this works. And we're just turning around, and governments realize we need to change something, but it's slow. So, for example, if you think about the economics of drug discovery, I haven't talked about this much, but this is important for you to understand. Is the early phases you spend relatively little money. And then in phase 3, you need to spend a lot because the trial is large and it's pivotal. But you also need to spend money through branded life. This is like getting